So this plant growth and responses to the environment, there's a lot to this. So we're just going to hit the highlights and think about some of the more applicable things to think about for commercial production of plants. And so some very basic terms, just thinking about plant growth, we're thinking about an irreversible gain in volume or weight. And there's really two ways that this can happen. It can be cell division, where you just have more cells getting added to the plant body, but it can also mean cell enlargement. That often gets overlooked. So plant cells do have some ability to shift their size, and they'll, eventually get to a mature size. And so cell enlargement is another thing to think about in terms of plant growth. So often you'll hear plant growth and development all in one phrase like that, but technically plant development is the progress of that plant through its life cycle. Whoops. So the picture on the right is showing that the plant on the right, yeah, there's more growth there compared to the one on the left, but the left is more developed because it's further along the life cycle. It's already starting to form flowers and eventually fruit. And so technically growth and development really are two different concepts in this realm. And you certainly have a lot of genetic determinants, what's going on with the chromosome, what genes are there. Certainly that is going to play a large role, but plants are very plastic in the sense that they can respond to their environment in a very fluid manner. And so the environmental, nature versus nurture type idea goes for plants as well as for animals. And so I think you could easily come up with this list yourself if you just think about some of the things that we've talked about in terms of what impacts the plant. And this is not exhaustive, just kind of giving you the examples. So we have, when we think about plants and how they're responding to their environment, sometimes they do that through a growth response. And so we'll talk about that. But one thing we need to think about with plants is, they really do have a way to move. Although at first blush that may sound silly, but really they do move in a purposeful way. And so this was actually studied originally and brought to, the public's attention back with Charles Darwin and his son Francis. They were really interested in how some of the more dramatic plants go through their movement, and they wrote a whole book on it. So, but that begs the question, how do you move if you don't have muscles? We have muscles that makes us have that ability to move, but plants do not have muscles. So what are they doing? So there's basically two big concepts. One is turgor movement, where you have a shift in water pressure, essentially, in certain parts of the plant body, and that will cause the plant to move. And so you're looking here on the bottom image are the stomata and the guard cells. So those kidney bean shaped guard cells, when they have water rushing into them, they actually will bend away from each other and reveal the pores. So that's a turgor movement. As water exits out of the guard cells, then the guard cells start to move closer to each other and close down the pore. So that's a really great example. And then it's basically just looking at the changes in water pressure. Off to the right is a leaf petiole. So that's the little stem of the leaf. Okay, so they cut off the leaf blade. You're not seeing that. But basically, many plants have this so-called pulvinous tissue at the base of their leaf petiole. And that, again, can swell with water. So there'll be a rush of water into portions of that and cause swelling, and that can allow the petiole to move. And the benefit there is that if the petiole has the ability to move and adjust, then it's adjusting its leaf blade so that the leaf blade can be, ideally plants like to have their leaf blade 90 degrees to incoming sunlight. So if it's high noon and the sun is coming straight down, you want the leaf blade kind of horizontal to the ground. So it's getting a 90-degree hit of sunlight. And so often with I'll show you some examples. Often it's an asymmetrical thing where, like on the case of the pulvinus, one portion of the pulvinus is swelling up and getting good pressure and the other part isn't. And that's what's allowing the petiole to kind of tilt and move in certain directions. So turgor movements are a big thing, and it can be quite rapid, so that it's pretty easy to get water to rush in and out of an area they just have to do a few things really to get that to happen. And so often you have turgor movements, and they can respond fairly quickly to something that's happening in their environment. Sometimes it's by some other organism perhaps touching them, and then they will move through these turgor movements. So sometimes it's just sunlight striking their body and they'll respond, like with the pulvini. That's plural for pulvinus. That's that tissue at the end of the petiole. In the extreme case here, the pulvinus area is causing very rapid movement in the sensitive plant. And so this is Mimosa is the genus name. Mimosa pudica is the full name of it. And it's called the sensitive plant, or sometimes they call it the shy plant or something like that. And so when you touch the little leaflets, they really close up really rapidly. And that's due to turgor movements. You have very rapid shift in turgor pressure, and that causes the leaflets to fold up. And it's fun to play with. Just if you get a sensitive plant, don't play with it too much for too long, all in one sitting, because it's kind of tiresome after a while. So be careful about that. But so when they notice this and you touch the leaflets and the leaves collapse like that, they think that that's actually a strategy to try to defend itself against getting eaten. So they've shown video of like insects crawling on those leaflets, and the leaflets will fold up very quickly, and the insect will actually like tumble off the plant because it loses its footing. And then the show the insect just kind of like, well, what just happened? And then it'll start crawling away or it'll fly away. And so it is an effective strategy to get something off of the plant body just through that really rapid turgor. So here there's a blow up again of the pulvinus region at the base of the petiole, the leaf stem essentially. And then here they're trying to show the sensitive plant with all those little leaflets, and that you have at the base of each one, you have a pulvinus region too. And so you get this asymmetrical, one side gets very good water pressure or turgor, the other side doesn't, and that allows for the leaflet to kind of collapse together, confuse what it might be crawling on them. So the fancy word for that, by the way, is seismonasty. That's like a something's physically messing with the plant. So seismic, you think about earthquakes and the seismic, the machines that measure the seismic force of an earthquake. That's kind of the idea that there's some kind of external mechanical force that is the plant is experiencing. So they call this seismonasty. Flowers do this though too. So here, this is kind of cool. You see a flower where the stigma, the female portion, its job is to collect pollen, and you can see it's open on the left, ready for pollination, and on the right, it's closed up. And so again, this is a turgor movement. So what tends to happen is after this plant gets pollinated, the stigma picks up that signal, oh, I got pollen on me, and it will then close down to do the next step of fruit formation. And so that's another example of turgor movements. Here's some really dramatic turgor movements. The mimosa plant is what you're seeing here. And again, just touching it, just barely touching it, causes those leaflets to fold. And I think you've all seen some video footage at some point in your life with Venus flytrap capturing an insect, and those modified leaves snap shut really rapidly and successfully capture a lot of different insects. Again, that's a turgor movement. And then sundew plants, again, this poor hapless fly is stuck now. And then that sundew will kind of curl around the insect and both of these plants will then start to digest away the insect. They're going mainly for the nitrogen in the exoskeleton, and they like all the nitrogen. So this is the idea of seismonasty again, where the insects crawling on them, that mechanical physical action on the plant causes the plant to respond in these pretty dramatic mechanisms. So those are really good examples of turgor movements that can be very quick in time. Here's another one that happens more on a day-night rhythm. So here you're looking at this idea of nyctinasty. Nyctinasty means sleep. Nastic is movement. And so this happens with quite a few plants. Not every plant will do this, but many do. I got fooled by this greenhouse late at night. It was 11 o'clock at night, and I was in there in the summer. We were growing some bean plants for an experiment, and I walked in really late at night, and I saw all the bean plants looking like they do on the right here, with their leaves tilted down. And my initial reaction was, "Oh no, someone forgot to water them!" You know, I thought they were wilting, so I was in a bit of a panic. And so then I was feeling all the pots though, the soil was nice and moist. And then I remembered, oh yeah, they do have this nyctinasty. So at night, they will actually alter their leaf angle, essentially. And they think they might do that to let like any rain that accumulates kind of drip off. Because at night, if you get a lot of standing moisture on plant parts, it tends to promote fungal growth, which isn't like the bad fungi, the stuff that might want to attack the plant. And so they hypothesize that might be why some plants do that, to allow for kind of letting the humidity in the water roll off of it, so to speak. The pear plant, there's actually a plant called the prayer plant that will do this, except instead of bending down, the leaves kind of bend up, so it looks like praying, hands folded in prayer. And so that's why they gave it the nickname, the prayer plant. So at night, you would see the leaves pointing straight up, and kind of pressing toward each other. I bought a prayer plant once years ago. It never did that though. Like, did I get a mutant or what? I wanted it to do this, of course, so I could demonstrate it. Could never get it to, even like in the middle of the night, I'd get up and look at it, and it would still be just normal. So I don't know what was going on with that. So it's not maybe 100%. Every single individual might not do that. So certainly these turgor movements are allowing the plant to respond and behave to these different stimuli, whether it be an insect crawling on them or this day-night rhythm where you have light and dark, and that's signaling to shift their leaf movements. Okay, so turgor movement, that's a big one. The other big concept of how do they move is through growth. That's actually a way for them to move, to grow and continue to, like, you think, for example, about roots growing through the soil. We actually use the phrase that they'll forage for nutrients. They'll grow toward the nutrient patches in the soil. And that's allowing them to move toward, a nutrient source. And so this is going on mainly through cell division, just adding to the tissue. But you can also see cell elongation playing some kind of role in that as well. And again, often you'll see it can be very asymmetrical, and I'll show you some examples of that. So you might get growth on, for example, one side of the stem, but not on the other, and that allows for a very particular type of growth movement that's makes sense for what's happening to that plant at that time. I'll show you some examples. And so gravitropism and phototropism are some ideas. I'll show you some examples of that. So by the way, tropism, when you hear that term, it usually is referring to a growth movement. And so in that regard, it's a permanent movement. If you're adding cells, that's kind of a permanent thing. And so we'll say a tropism is a permanent movement because of some external stimuli. And it can either be toward or away from the stimulus, it just depends. And so we're thinking about this idea of, well, let me show you an example. So phototropism is this idea where you have light coming from one side, just shining on one side of the plant. This happens if you have a plant in your windowsill in your home. And then you can start to see the plant bending toward the window, toward that sunlight. And so what's actually happening here is that asymmetrical light, that unidirectional light, is causing auxin, which is right at the apical meristem. That auxin hormone will start to flow down, but flows only on the dark side of the stem and tells those cells to grow, grow, grow. And then that causes that bending pattern toward the light. So again, it makes sense if you think about it for a second, because those leaves want to get 90 degrees to the light coming through the window, right? And so it makes sense that they'll present their leaves as close to 90 degrees as they can. And so to do that, they got to bend their stem. And so if you take this light and you put it on the other side of the plant, eventually the plant will start bending back again. So you can actually do experiments. I think I might have provided some links to, oh yeah, I think this link down here, if you put that into your browser, it pulls up a video where they show someone doing that. They keep switching the light on the plant and you'll see the plant keep bending back and forth toward the direction of the light. And so I put Darth Vader here to get you to think about how the auxin is moving to the dark side. And so that's how I always remember this idea after watching Star Wars over and over when my boys were little. And so this is an example of asymmetrical growth, in this case, because of unidirectional light. And so phototropism, movement due to light, that unidirectional light. And so this is actually quite handy for plants because they can then track the sun through the day. So of course we have sunrise in the east, very low on the horizon, and then gradually the sun goes up to, the zenith at high noon, and then starts to descend again into the west. And so probably the most famous example are those sunflowers, where their actual flower heads can turn on using Turgor movements in this case. And then you can do experiments. This lower left one, it was a really cool experiment where they were showing that phototropism idea. And then what they discovered when they were doing this experiment is that this shaded plant, what they said was they turned off all the lights and then just watched to see what it would do. And the plant would reorient itself to face east. So through the night, it's like it knew through that long dark stretch, okay, I need to shift over so that my leaf blades are facing east and ready for the sun to come up. So that was kind of an interesting find when they did these kinds of studies in the lab, that they learned that plants are kind of able to keep track of time and figure out that they have to turn and be ready for the east sunlight to come up. So pretty cool. And again, so this response in this case with solar tracking is more of a Turgor movement, where you can get the shifting like this in response to the sun tracking across the sky. With this idea back here, this is more like if you have steady unidirectional light, okay, like if you have plants in your windowsill and you just leave them alone and they only get light coming from that direction all the time, then you'll get this growth response and it'll stay that way. They'll stay facing the sun coming through your window. Unless you decide to change them or shift the pot. You can turn the pot and have it face away, and then it'll slowly start to rebend toward the incoming sunlight. Okay, well, my timer just went off. So let me just see what the next. Okay, so yeah, we'll stop there. And let's see, before I forget, could you please go into EduCat and do the course eval for the lecture? I'll step out of the room for about 5 minutes to let you do that. And then I'm going to come in and we'll use the rest of the time to do the review. Okay, so the final exam starts with the slide set on photosynthesis, right? the light reaction. So we have essentially two slide sets on photosynthesis, the light reactions, and the next one is looking at how sugars get made. So the Calvin cycle. So I'm here to provide answers to any questions you may have and with regard to any of the content. There will be a fair amount of points being asked on these two slide sets. This is a really important concept. Yes.
We briefly go through like what steps we should focus on.
Absolutely. Okay. So when you think about some of these slides, some are more detailed than others. And so when you're zooming in, let me look at, okay. So, all right, so let me start with this one. This one you can see is pretty detailed. And so I put this here mainly to emphasize the idea that you have two photosystems that are both getting hit with sunlight energy. Okay, it's photosystem 2 and photosystem 1. They both have to get good quality light coming at them in order to excite those electrons in the chlorophyll pigment in the reaction center. So that's one of the main reasons I put this here. However, I'm not asking you to know all this detail in the image. What I want you to focus on is the subsequent slides where I type it out in words. So this is more general than what I just showed you. And then if you look at this slide as you are then reading through what I've typed up here, that's the way to study this. Okay, so I've kind of zoomed out from that really detailed figure, and I'm giving you more of the highlights rather than all the details. So I would put that image up to refer to back and forth, but pay attention to my typed up, like this is what is going on with the light reactions. And so here you're looking at the idea that you have those two photosystems and that they are put together in a series or a chain. And so this is kind of like the big picture, what is photosynthesis going on? You have the use of light, and you need that in order to strip electrons off water, which is incredibly resistant to giving up electrons. And as you're doing all that, you see that one of the big payoffs is those electrons finally end up over at NADP+. All right, so going back here, you see that's the ultimate electron acceptor, NADP+. And so when you end up with NADPH, that's great. That's a really good thing for the plant to be able to have handy. All right. So let me go to the next. So this is just trying to orient you to what are those photosystems, and that you have those special chlorophylls in the reaction set. And that's where we really see, the chemical reaction occurring. And then just getting to know the players. So you have the big complexes, the two photosystems. And then this is a huge transmembrane complex as well as this. So those are like the big players in the process. All right, so we call them transmembrane or integral membrane proteins. So I want you to know the names of those. And basically, we're looking at the idea that when light strikes the photosystem, what really kicks it off, we usually start at photosystem 2 and say, okay, what happens when light comes in and that light energy gets to the reaction center? You actually get a shift of electrons from one molecule to the next. And that's really important because that's what kicks off the electron transport chain process. And so they make a big deal about this, that P680, that's the reaction center chlorophyll molecule, is giving off an electron, and it gets donated off to this acceptor that we call FEO. feel fighting. Okay, so that step is important because that's what kicks off the electron transport chain. The other really big idea here, that as this is happening, part of that light energy is used to extract electrons off of water. So that's why we call water the ultimate electron donor, because that's the ultimate source of where, you know, once this chlorophyll in the reaction center loses an electron, then it's like, I've lost an electron. I need to get that replaced. And so it turns to water for that. All right. So some of the energy through, you know, the light energy is being used, and it's in this oxygen evolving complex. That's what they named it, but that's the area inside the photosystem that strips electrons off water. So these typed up ideas that I have are the level of detail that I'd like you to know. And okay, let's see, let me get out of this. Out of that. Okay, so looking through these slides, what other questions do you have on the light reactions? By the way, so as this is all happening, so this is showing, the dotted line is showing the electron flow ultimately from water all the way to NADP+. So the other big concept, after we're done thinking about, okay, that's how the plant is making the NADPH, but as this electron transport process is happening, it's also allowing for protons to be building up in the lumen. And that's another really important concept, because what it does then, that proton gradient is then a way for the plant to make ATP. So there's really two huge concepts when you look at this picture. You have the electron transport chain going, which allows for NADPH. But as that electron transport chain is going, it sets up the conditions to allow for a proton gradient to get established. And that's how the plant makes the second big chemical that it needs, which is ATP. So the fancy name for that is linear photophosphorylation. That's what they are referring to in terms of making ATP. Photophosphorylation means light energy, photo. Phosphorylation just means ADP is getting phosphorylated. All right, so don't get tripped up on this. And I will be referring to this idea as linear photophosphorylation on the exam. So don't get tripped up on this phrase. It's just talking about how does the plant make ATP, all right? And the linear portion of that phrase is just referring to the idea that you have that linear flow of electrons. And the way that that is designed, it allows for a proton gradient to get established. So linear photophosphorylation. So be familiar with that term, because that's how I'll refer if I ask about, you know, basically I'm asking about how does the plant make ATP through the light reaction. Two huge goals. Yeah. Big picture ideas. You bet. Any other questions on this slide set? Well, let's go into the next slide set, which is the how does the plant make its sugars? So then once the NADPH and the ATP get made, that's still, all happening in the chloroplast and it's happening in the stroma. And that's good because when those ATP and NADPH gets made in the stroma, you also have the Calvin cycle cooking along, hopefully. So here they're trying to link the two parts of photosynthesis together, where products of the light reactions are being used to drive the Calvin cycle or the C3 cycle. And so here we were looking at the idea of how does the plant bother, how does it go through the process to make its sugars? And that's the idea of taking CO2 in through its stomata. And it's got to create this C3 cycle in order to get enough building blocks essentially to eventually make either sucrose or starch. And so this is hopefully cooking along all day long. And so the CO2 is going to react with RUBP, which is a 5-carbon sugar. Okay, that's a 5-carbon sugar. And a very special enzyme facilitates that. What's the enzyme acronym? Do you remember? Rubisco. All right, so make sure that you don't get these two confused, please, please, please. Rubisco is the enzyme. RUBP is the 5-carbon sugar. Very, very different. Rubisco, the C stands for carboxylase function, and the O stands for oxygenase function. All right, so, whoops, where'd it go? Not that one. Oh, okay. So the 3 stages here, I do need you to know those in order. Like first you do carboxylation, where CO2 is going to react with RUBP. Then the next big thing is reduction. Then the last step is regeneration. You need to know those 3 parts of the so-called pie chart process of the C3 cycle. I also want you to know where are ATP and NADPH getting used. That's kind of important to see how the light reactions work into, the products of the light reactions are working into helping with the C3 cycle. So during the reduction step, you have both ATP and NADPH are needed. And then in the regeneration, it's only ATP. And then you can see carboxylation is good. It doesn't need any help. It's good to go. As long as Rubisco sees a bunch of CO2 coming in, it will just automatically react it with RUBP. No special help required. So again, the words that I've written out, those are the steps that I think are important to think about. Let's see. Let's go this way. Oh, yeah. Come on. Oh, just go. All right, let's go back to this view. Okay. So what else on this? Any other questions on this slide set? Remember, we have the PCO issue to deal with, because Rubisco sometimes doesn't see enough carbon dioxide. Instead, it'll start to take oxygen. And that's not such a good thing. So if oxygen comes in instead of CO2, then you get that PCO cycle, which is that. It's the one that has those 3 different organelles, and it's complicated and only partially successful at getting some more. Ultimately, the good thing about it, I guess, is that you do see, ultimately, you can build some more PGA. So that's good. But you do end up, at the end of it, you do get another PGA molecule showing up. But at the expense, I wish the artist had been a little bit more blatant here. The CO2 coming off is just lost. It's just gone. The plant can't capture that in any way. So that's a definite negative to the PCO. But the upside is, through all this mess, it does end up making another PGA that can feed into the calcium. Other questions on this slide set? What did we do after the light reaction? What was the next thing? Oh, C4. C4 and CAM. So again, this is like, okay, some plants are faced with environmental regions of the world where it might create situations where CO2 isn't coming in as much as it should into Rubisco's active site. And so oxygen has the risk of getting in there too much. And so these C4 and CAM plants have evolved mechanisms to deal with that in order to minimize, basically, they're trying to minimize the PCO cycle. And so again, it's just a way, ultimately both of these, the C4 and the CAM, it's just a way for the plant to make sure that that Rubisco enzyme is kind of just bathed or flooded with a lot of carbon dioxide and very little or no oxygen. And even if it's hot out or really, really hot out or drought time period, they're still good at minimizing oxygen coming into Rubisco's active site. Questions on the C4 or the CAM? So they're showing they're showing the CO2 coming in. Okay. And they're saying it reacts with phosphoenolpyruvate. So does Rubisco do that step? No, that's not Rubisco. So when you look at the C4 and the CAM, Rubisco is not the immediate enzyme that will work on the CO2 that's coming in to the plant. Instead, it's that pepcase enzyme, which is way more efficient in the sense that it only cares about carbon dioxide and it won't bond at all with oxygen. So that pepcase enzyme is the one that's waiting to greet CO2 as it comes in from the stomata. Case is carboxylase, is short for carboxylase, which just means that you're taking CO2 and reacting it. When you carboxylate something, you're sticking a CO2 onto some kind of molecule. In this case, it's PEP, phosphoenolpyruvate. Any other questions? The C4 and CAM have a lot of the same players. It's just but they've, evolved very different mechanisms, right? So the C4 have a difference in physical location of where things are happening. It's the bundle sheet versus the normal mesophyll cells. And then the CAM plants, they just do a day-night rhythm. So they have a temporal segregation of when things are happening. CAM plants are really, really good at conserving water because of how they do it. Okay, what was next after this slide set? Blinking at the moment. Mineral nutrition. Thank you. So when you're studying this content, mineral nutrition, think about the soil characteristics. Well, those are important because that's really setting the stage for how the minerals become available to the plant. You have that negatively charged surface on the soil. And so that really does limit what minerals are like held in the soil. It's only the cations that are there. All the negative ions, the anions, they just stay in the water and they can percolate past the root zone, certainly. So there's no reservoir for the anions. So think about the soil chemistry and like how that functions. And then as far as the actual minerals, please know, where did it go? So the immobile versus mobile, I do want you to know that in the sense of once the plant takes it up, can they redistribute those minerals or not? That's what that mobile versus immobile. pH range of soil is important to think about. And then we went through some of the minerals, not all of them. So the ones that are in the PowerPoint, remember with the textbook readings, you only use the textbook to back up what you're learning from the PowerPoints, okay? So if something's in the chapter that I did not talk about, you don't need to know it. So for sure, the best way to study for this final is to pull up the PowerPoint. That's your study tool. And only use the textbook if you're confused by something you see on the PowerPoints, okay? So I haven't finished making it yet, but I'm guessing it's going to be at least 125 points. And there will be more multiple choice than there will be essay for sure. So it'll be similar to what we've already had in the sense of multiple choice portion, essay portion. I'm guessing the essay might be 4 questions again, roughly 4 questions, some shorter or some longer. So I put the drag and drop. I can even put that multiple choice portion for and essay portion. But thank you for reminding me. There probably will be drag and drop. You done that yet? All right, so we're probably 4. We're gonna get a word bank, and you'll either match or put drag and drop function. Okay. So on Friday, I will do a little bit more talking on plant growth and responses, and I'll also allow time to have some other questions. We can finish up doing a review on Friday